today's session. In the previous class, we basically discussed about the types of proteins which are present in membrane, and then we discussed about their major functions. I want to discuss one of the major function, which is transportation across the cell membrane, as this is the function which helps in maintaining the cell viability. And why I am saying this, for example, you know that in cell a lot of biochemical reactions are taking place all the time, right? So if a biochemical reaction is taking place, there will be substrate and there will be products, and then there will be byproducts also. So if these byproducts are not excreted outside the cell or are not taken care of, they might lead to the cytotoxicity of the cell, correct? Hence, to assure that the cell has adequate nutrients and that cell is maintaining its viability by maintaining its physiological conditions, there are transporters or proteins in the membrane which basically works in transporting solutes across the membrane, right? As you can see in the C figure here, correct? So there will be transporters. So if I'm saying that there's a transporter which is transporting solutes across the membrane, to which category or type of protein they will be? Let me be a bit more clear. Yeah, so these were the different classes of or types of proteins which are present in the membrane, right? Peripheral protein, amphitropic protein, integral protein, lipid linked proteins, correct? So now I am saying that there are proteins which are involved in transporting substrates or solutes across the membrane. So to which category, first, second, third, fourth, to which category ideally these proteins or transporters should belong to? Should they be of peripheral type protein? Should they be amphitropic proteins or integral proteins? And even in integral protein, will they be endo-ecto type of proteins or transmembrane proteins? And if none of the above, then will they be lipid linked proteins? And in lipid linked proteins, the major type was GPI linked proteins. Okay, so I want you guys to think, if I need to transport across the membrane, will the peripheral type of protein work? The answer will be no, right? Because peripheral proteins will be associated to one leaflet of the membrane. They won't be transversing the membrane so that they can carry the solute or substrate across the membrane, right? Hence, the first option is not right. Similarly, amphitrophic protein is also a type of peripheral protein at the end of the day, and so this option is also out. If we talk about lipid linked proteins, even they, though they are associated with the membrane deeply, but the protein doesn't transverse the membrane, correct? If you remember the cartoon representation of lipid linked proteins, there's a lipid linkage which is embedded in the membrane and the protein is still on the surface of this linkage. Hence, it is not transversing the membrane. So a solute or substrate to pass across the membrane, there should be a protein which is completely transversing the bilipid layer from one end to another end, correct? Hence, the category to which transporters belong to will be integral type of protein, correct? And even in integral type of protein, it will be transmembrane protein. So if you remember the discussion about transmembrane proteins, this is the representation of a membrane, and for a protein to be transmembrane, it has to span both of these leaflets, correct? Hence, we can say that transporters will belong to the transmembrane type of integral membrane protein. Done. And you can see the similar thing in the representation also, that they are transversing the membrane and hence they are transporting the solute from one side to the another side depending upon the requirement and the type of transportation we are looking into, correct? So why do we actually need a protein to transport across the membrane? Why can't the membrane itself transport substrate or solutes? So if you remember our earlier discussions when we talked about lipid being amphitrophic, hydrophobic, so there has to be something in the membrane to provide the transportation of substrates or solutes which are hydrophilic in nature, right? Because of the energy barrier faced by the polar molecules when they enter into the highly hydrophobic or non-polar environment. So this is a very brief representation of such solutes. So if you look at gases are permeable across the membrane and they can directly be transported from outside to the inside via the lipid bilayer itself, and they necessarily do not need dedicated proteins or transporters for their transmission. Okay, if we talk about small uncharged or polar molecules, so some of the uncharged polar molecules, because they are smaller in nature, they can permeate through the membrane, but the permeability is slightly less. So if there is a requirement in the cell and it requires a huge influx of suppose water or urea or ethanol, then mere diffusion across the membrane will be a slow rate, and hence the cell might suffer the consequences if the need is not fulfilled in the required time. Though it can transport, but still the permeability is kinda less because of the energy barrier. If we talk about large uncharged polar molecules, then definitely they'll be non-permeable across the lipid bilayer, and they require assistance of proteins and dedicated transporters so that they can be taken up inside the cell or outside the cell depending upon the requirement. Okay, then ions. So these are small but they are charged molecules at the end of the day, correct? So even the charged molecules, they will be non-permeable to the membrane because of the energy barrier, correct? Then charged polar molecules, definitely there is no way that these molecules will be able to transport across the membrane without the assistance of specialized proteins or areas inside the membrane which provide the hydrophilic environment for the transportation of these molecules. Okay, so this gives us some brief idea that why do we actually need membrane proteins, especially the transporter type of proteins, because cell require all this. You can see that CO2, N2O2, there are many biochemical pathways in which these are the raw materials, correct? Similarly, ethanol, water, urea, glucose, fructose, small ions such as potassium, magnesium, calcium, chlorine, then amino acids, ATP, all these are basic requirement of a cell to survive and do biochemical reaction. So if they are not being transported inside, for example, if they are being used as a substrate or vice versa, for example, if they are produced as a consequence of a chemical reaction, they need to transfer or transport or travel to the required places, right? And even the to and fro motion of these substrate requires dedicated proteins in the membrane so that they can be easily transported. Okay, so if you talk about the membrane transport, basically they are of two types: passive transport and active transport. In passive transport, basically there is no association of energy. It basically transports across the membrane via concentration gradient. Higher the concentration of a substrate, it will gradually move towards the place where the concentration is less spontaneously, right? So in passive transport, there are two types of passive transport: simple diffusion and facilitated diffusion. In simple diffusion, as you can see in the representation itself, that the small molecules will directly transfer across the membrane without the assistance of any protein and energy, right? So the example will be of gaseous substrates, correct? Now if we talk about facilitated diffusion, then in facilitated diffusion, though the transportation is across the concentration gradient, but it requires the assistance of protein, so it is called facilitated diffusion. The second type of transportation is active transport. In active transport, there is association of energy. It can be direct energy via ATP, or it can be indirect energy of another substrate, right? So if there is association of direct ATP energy, if there is association of direct ATP with the protein, it is called active type of transportation or primary active type of transportation. If there are substrates, for example, if you can see in the representation itself, there is one substrate which is high in concentration, there's another substrate which is less in concentration. So what will happen is there will be transportation against the gradient of this less abundant substrate by using the energy associated with the substrates which are higher in number. So in this, there is no direct association of ATP, but the energy utilization is still there. Hence, it will be called as secondary active transport. Another major difference between passive transport or active transport is this. Passive transport, as I have mentioned earlier, that it depends upon the concentration gradient. Hence, it will always be from high to low, meaning wherever the substrate is higher, it will always passively transport it towards the across the membrane wherever the concentration is less, correct? In other words, passive transport is concentration dependent, right? On the other hand, in active transport, the transportation is actually happening against the concentration gradient, meaning, for example, if you already have a lot of substrate here, right, then why would passively, without the association of energy, you will transport more substrate from this side to that side? Because it will be against the concentration gradient, correct? So for this you need energy, and similarly in active transport this is what happens. As you can see that the substrate is less here and more here, correct? Yet the substrate is being taken from this side to that side via the ATP hydrolysis. So working against the concentration gradient will require energy. The lamest example or the daily example which you can see is your water pump, correct? Everyone has a water pump. So what we are doing is we are pumping the water against the concentration gradient or against the gravity, right? Hence we need to pull or push the water outside from the ground itself. We need to push it, we need to provide energy for doing it. Similar thing is happening here itself, that energy is associated for the transportation of a substrate which is already in lesser concentration, and it is being transported from lesser concentration to across the membrane where the concentration is comparatively more. Hence we do require energy. To understand better and have a brief idea about the transportation, I have this brief video. Let's see and have an overlook of how we can understand transportation and
membrane transport,
their basic properties. So let me share the screen. 1 minute.
So how do substances move into and out of a cell? Well, cells have a plasma membrane that acts like a fence, and this plasma membrane is what regulates what moves into and out of a cell. The membrane is comprised mainly of phospholipid molecules and has protein molecules scattered throughout its structure. The phospholipid molecules have a polar hydrophilic water-loving head and two nonpolar hydrophobic water-fearing tails. The membrane is formed as the heads face out and the tails face in, forming a bilayer, as you can see here. The inner part of the cell, called the cytosol, is here, and the outside of the cell is surrounded by a fluid called interstitial fluid. Substances move into and out of a cell through several different processes called membrane transport. There are two main processes: passive transport processes and active transport processes. The main difference between the two is that passive processes do not require energy expenditure, and active processes do require the cells to expend energy. Let's start by looking at the passive processes, which include simple diffusion, facilitated diffusion, and osmosis. Diffusion is the movement of a substance from where it has a high concentration to where it has a low concentration, or the tendency of a substance to spread out evenly over a given space. For instance, when a sugar cube is dissolved in water. Over time, the cube will dissolve and eventually spread out evenly in the water until it reaches equilibrium. Diffusion occurs down a concentration gradient, which is a difference in concentration of a substance between two areas. So the sugar molecules will move from an area of high concentration to an area of low concentration. Cellular diffusion is when diffusion of a solute, which is a dissolved substance, occurs across the plasma membrane from an area of high concentration to an area of low concentration. This is dependent on the concentration of a substance in the interstitial fluid outside of the cell and the cytosol inside of the cell. This can occur through simple diffusion or facilitated diffusion. Simple diffusion occurs with solutes that are small and nonpolar. By being nonpolar, they can move in between phospholipid molecules that form the plasma membrane because the interior region of the membrane is nonpolar. Some of the materials that move by simple diffusion include the gases oxygen and carbon dioxide and small fatty acids. So if there's a higher concentration of oxygen O2 molecules outside of a cell, they can move down the concentration gradient across the membrane without assistance and into the cell as long as the concentration gradient exists. And if there's a higher concentration of carbon dioxide CO2 molecules inside a cell, they can move across the membrane without assistance out of the cell into the interstitial fluid. So again, simple diffusion is when nonpolar molecules pass directly through a membrane. The second type of diffusion is facilitated diffusion. This applies to solutes that are small and either charged or polar. Because these solutes are polar, the nonpolar phospholipid bilayer blocks them from passing through the membrane and into or out of the cell by simple diffusion. However, they can pass into and out of the cell with the assistance of plasma membrane proteins through a process called facilitated diffusion. There are two types of facilitated diffusion: channel-mediated diffusion and carrier-mediated diffusion. The difference between the two is the type of transport protein used to move the substance across the membrane. Channel-mediated diffusion is when an ion, which is a charged particle where its total number of electrons does not equal its total number of protons, giving it a positive or negative charge, moves across the membrane through a water-filled protein channel. Each protein channel is typically specific for one type of ion, and there are two types of channels: a leak channel, which is continuously open, and a gated channel, which only opens due to a stimulus and only stays open for a fraction of a second. So for a sodium positive ion, it can pass through a sodium positive leak channel continuously, and a gated sodium positive channel will only open due to a stimulus to allow the ion to pass through into the cell. Carrier-mediated diffusion involves the movement of polar molecules, such as simple sugars or simple carbohydrates, and amino acids across the membrane. This is accomplished by a carrier protein, which actually changes shape in the process. For instance, glucose binds to a carrier protein, which changes shape and moves the glucose molecule to the other side of the membrane. Now for osmosis. Osmosis is the passive movement of water through a selectively permeable membrane. This occurs when there's a difference in concentration of water on either side of the membrane. This can happen in one of two ways. Water can slip between the phospholipid molecules that make up the plasma membrane, or through integral protein water channels that are called aquaporins. The plasma membrane is not permeable to most solutes, such as charged, polar, and large substances. So for example, one side of the membrane, the cytosol or interstitial fluid, can have more solutes than the other side. Let's say the interstitial fluid has 3% solutes and 97% water, and the cytosol side has 1% solutes and 99% water. In this example, water will move down its concentration gradient from the 99% cytosol side to the 97% interstitial side to achieve equilibrium. Now let's look at active processes. Active processes require the use of cellular energy for membrane transport. There are two types of active processes: active transport and vesicular transport. Active transport is the movement of a solute against its concentration gradient or going from an area of low concentration to a place of higher concentration. Vesicular transport is the transport of large substances across the plasma membrane by a vesicule, which is a membrane-bound sac filled with materials. Active transport has two types: primary active transport and secondary active transport. In primary active transport, cellular protein pumps, called ion pumps, move ions across the membrane against their concentration gradient. Let's take a look at an example. Here we have a membrane along with a sodium positive and potassium positive protein pump. 3 sodium ions and ATP bind to the pump, ATP being like fuel or the form of cellular energy. In order to release that energy, ATP breaks down into ADP and P. P binds to the pump, and the release of energy causes the pump to change shape and release the sodium positive ions into the interstitial fluid. 2 positive potassium ions bind to the pump, and the P produced by the ATP is released into the cytosol of the cell. The transport protein, or pump, reverts back to its original shape, releasing the positive potassium ions into the cytosol, and the pump is ready to do its job again. In secondary active transport, a substance is moved against its concentration gradient by using energy provided by the movement of a second substance down its concentration gradient. So, a substance that is moving from a place of high concentration to low concentration will provide the energy to move a second substance from a place of low concentration to high concentration. There are 2 types of secondary active transports: symport, where 2 substances are moved in the same direction, and antiport, where 2 substances are moved in opposite directions. In our example here, we have positive sodium ions moving from high concentration outside of the cell to lower concentration inside of the cell. A symporter will use the energy created by this to move a glucose molecule from a low concentration area to a high concentration area. And here we have an antiporter, again using the energy from moving positive sodium ions from high concentration outside of the cell to lower concentration inside of the cell, and H positive is moved up its gradient and outside of the cell. Vesicular transport involves the transport of larger substances, such as proteins or large carbohydrate polysaccharides, across the plasma membrane. There are 2 types of vesicular transport: exocytosis and endocytosis. In exocytosis, materials are secreted from the cell to the interstitial fluid outside of the cell. Typically, membranous vesicles, formed by the Golgi apparatus, transport materials to the cell's membrane. At the membrane, the phospholipid molecules that make up the vesicle fuse with the phospholipid molecules that make up the plasma membrane, and the contents of the vesicle are released outside of the cell. The vesicle membrane now becomes part of the plasma membrane. In endocytosis, the plasma membrane kind of traps the substance by folding inward. The lipid bilayer then fuses to form a vesicle surrounding the substance. There are 3 main forms of endocytosis: phagocytosis, pinocytosis, and receptor-mediated endocytosis. In phagocytosis, a large particle is engulfed by the newly formed vesicle, and this vesicle fuses with a lysosome, which is a membranous vesicle that contains digestive enzymes that break down the particle into its component molecules. Phagocytosis is also called cell eating. Pinocytosis, which is also referred to as cell drinking, is when the plasma membrane folds inward and engulfs droplets of interstitial fluid that contain dissolved solutes that can be used by the cell. Receptor-mediated endocytosis involves using receptors on the outside of the plasma membrane. These receptors bind with molecules in the interstitial fluid, and the membrane folds, enclosing the receptors and the bound molecules, to form a vesicle for transport within the cell. One last thing to note: both vesicular transport processes, endocytosis and exocytosis, require energy. And that be the basics on transport across.
Right, so that was the overview of the transportation across the membrane. Okay, so now you have basic idea about the types of transportation across the membrane. So we will look at some of the properties which are associated with these type of transportation. So we have briefly talked about simple diffusion, facilitated transport, active transport, and co-transport, right? So in simple diffusion, if we talk about whether they require a specific type of protein or not, so we can say that in simple diffusion there is no association of any type of specialized proteins, right? It is simply happening across the membrane via the simple diffusion, correct? While in facilitated transport, though it is happening along the concentration gradient, but it does require specialized transporters to be present in the membrane for the transportation. Similarly for active transport and co-transport. Solute transported against its gradient. So as we have mentioned that in passive transport, the transportation occurs along the concentration gradient, while in active transport the transportation occurs against the concentration gradient. Hence for simple diffusion and facilitated transportation, there will be transportation across the concentration gradient, and for active transport and co-transport the transportation will be against the concentration gradient. Okay, coupled to ATP hydrolysis. So we know in active transport, and even in active transport, only primary active transport is the one in which there is direct hydrolysis of ATP is associated. In other types of transportation, especially active type of transportation, direct ATP hydrolysis is only associated in primary and not in secondary active transportation. And in passive, there is no discussion of having energy association for the transportation. Then driven by movement of a co-transported ion down its gradient, then that is present only in secondary active transportation and not in any other type of transportation. So the examples will be simple diffusion will be small molecules and gaseous compounds. For facilitated transportation, it will be glucose, amino acids, ions, and waters. Don't worry about unipolar and channels. We're going to discuss it in the next slide itself. Then for active transport, the transportation happens, the substrate which are actively transported are ions, small hydrophilic molecules, lipids, ATP power pumps, via ATP power pumps, etc. Co-transportation again for glucose, amino acids, and various other carbohydrates. Okay, so if we are talking about transporter, so there are different types of transporters in the membrane. Basically 3 types. First, channel, second, transporter, third, ATP powered pumps. So in channels are basically gates in the membrane. Okay, so gates are just a passage in the membrane through which the transportation occurs along the concentration gradient. Then comes the transporters. Transporters can be of 3 types: unipolar, sympolar, and antipolar. In unipolar, there is one substrate which is being transported across the concentration gradient. Second is the sympolar. In this, 2 substrates are transported simultaneously, but one is along the concentration gradient and the other is against the concentration gradient. Then we have antipolar. In antipolar, there is transportation of solutes against the concentration gradient, but both of these are transported in different directions, right? And then we have ATP powered pumps, in which the transportation of substrate occurs against the concentration gradient with the help of ATP hydrolysis, correct? Then these are some basic transporters. For example, if we are saying that small molecules or ions are transported across the membrane both via channels and transporters, then we need to understand that all these transporters in the membrane are working simultaneously to maintain the gradient in the cell. So if you look here, the gradients are indicated by triangles here. So this indicates that sodium is more here and potassium is more inside the cell. Okay, with the tipping point towards a lower concentration. The sodium potassium ATPase in the plasma membrane uses energy released by ATP hydrolysis to pump sodium out of the cell and potassium inwards. This creates a concentration gradient of sodium that is greater outside the cell than inside, and one of the potassium that is greater inside than outside. Movement of positively charged potassium ions out of the cell through membrane potassium channels create an electric potential across the plasma membrane. The cytosolic phase is negative with respect to the extracellular phase. A sodium lysine transporter, a typical sodium amino acid co-transporter, moves two sodium ions together with one lysine from the extracellular medium into the cell. Uphill movement of amino acid is powered by downhill movement of sodium ions, which in turn is powered both by the outside greater than inside sodium concentration gradient and by the negative charge on the inside of the plasma membrane, which attracts the positively charged sodium ions. The ultimate source of energy to power the amino acid uptake comes from the ATP hydrolysis by the sodium potassium ATPase pump. Since this pump creates both the sodium ion concentration gradient and via the sodium channel, the membrane potential which together powers the influx of sodium ions. So you can clearly see how beautiful they are in synergy, and they are maintaining the concentration of one ion to another, which leads to the transportation of a amino acid from one side to the another side. So all these active transportation, passive transportation channels, all these work together to maintain the cell's concentration gradient across the membrane. So if we look at the overview of the transmembrane protein, cellular membranes regulate the traffic of molecules and ions into and out of cells and their organelles. The rate of simple diffusion of a substance across a membrane is proportional to its concentration gradient and hydrophobicity. With the exception of gases and small uncharged water molecules, most molecules cannot diffuse across a pure phospholipid bilayer at rate sufficient to meet the cellular needs. Membrane transport protein provide a hydrophilic passageway for molecules and ions to travel through the hydrophobic interior of a membrane. Three classes of transmembrane proteins mediate transport of ions, sugars, amino acid, and other metabolites across cellular membranes: cells, transporters, and ATP powered pumps. Channels form a hydrophilic tube through which water or ions move down a concentration gradient, a process known as facilitated transport. So I guess we'll end our class today here itself, and we're gonna discuss about another type of transporter itself, the membrane one, and the ATPase, sodium potassium ATPase, and then we'll discuss something more about the transportation, and then I guess we'll wrap up our first module and we can go to our second module maybe by next week. Feel free to ask any questions if you have, and you can mail me as usual. Thank you for your attention.